then okay, so there are two types of nitrogen based purines, that is adenine, guanine, pyrimidine, that is cytosine, uracil and thymine. So cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA, right? At the place of thymine, right? Nitrogenous base is linked to the OH of 1 dash carbon pentose sugar through the N glycosidic linkage to form a nucleoside. Now adenosine deoxy, adenosine, guanosine deoxy, guanosine, cytidine deoxy, cytidine, uridine or deoxythymidine. And phosphate group is linked to OH of 5 dash carbon of a nucleoside through phosphoester linkage, and they form it's like nucleotide. Okay, or deoxynucleotide depending upon the type of sugar present. So two nucleotides are linked through 3-5 phosphodiester linkage to form a dinucleotide, right? Okay, so at the 1 dash OH it's like the nitrogenous base, and at 5 dash it's like 5 dash OH, which is phosphate group, and 5 that is 5 dash and 3 dash, so they form the phosphodiester linkage. Okay, 5 dash and 3 dash, right? So a polynucleotide change, right? So okay, so a polymer thus form has at one end a free phosphate moiety at 5 dash end of sugar, which is referred to as 5 dash end of polynucleotide. Similarly, at the other end of the polymer, the sugar has a free OH of 3 dash carbon group, which is referred to as 3 dash end of the polynucleotide chain. Now the backbone of a polynucleotide chain is formed due to sugar phosphate. Then the nitrogenous bases linked to sugar moiety project from the backbone. The nitrogenous bases linked to sugar moiety project from the backbone. Okay. Now in RNA, every nucleotide residue has an additional OH group present at second dash OH in the ribosome. Okay. Thymine, chemical name, that is 5-methyl-uracil. Okay, DNA was an acidic substance present in nucleus was first identified by Friedrich Mister in 1869. So it is nuclein. In 1953 by James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. Okay. Now the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. So this one based on the Irving Chargaff, right? Irving Chargaff. For a double stranded DNA, the ratios between adenine and thymine. Okay, the ratios between adenine and thymine. So ratios between the adenine and thymine. Okay. Okay. So Irving Chargaff, that for a double standard DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equal 1. Are constant and equals 1. Okay. Now the salient features of the double helix structure of DNA are as follows. So it is made up of like two strands are paired through. No, it is made up of two polynucleotide chains, where the backbone is constituted by like sugar phosphate, and the and basis project inside. So sugar and phosphate. So the two chains have anti-parallel polarity. It means if one chain has polarity, that is 5 dash to 3 dash, the other has 3 dash to 5 dash. Now adenine and thymine has two hydrogen bonds, and cytosine and guanosine has 3 hydrogen bonds. So as a result, like always, a purine comes opposite to a pyrimidine. So This generates approximately uniform distance between the two strands of the helix. Now two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer, and they have like 10, and they have roughly like 10 base pairs in each turn. Okay, so consecutively the distance between a base pair in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bond, confers stability to the helical structure, right? So you say adenine, thymine, guanine, cytosine. Since this is DNA double helix, right? So double standard polynucleotide chain. You see, and then like now the central dogma given by Francis Crick. The genetic information flows from DNA, RNA, and protein. Okay. So DNA to DNA, that is replication, to DNA to mRNA, that is transcription, and from mRNA to protein, that is the translation. So in some viruses the flow of information is in reverse direction, that is from RNA to DNA, and then DNA to RNA, then protein. So that is from RNA to DNA, that is the reverse transcriptase. Okay, the packaging of DNA helix. Now 2.2 meter is the length of the DNA, and that has the like human genome has like 6.6 into 10 power 6.6 into 10 power 9 base pairs, and the distance between the two consecutive base pairs is 0.34 into 10 power minus 9 meter per base pair. Okay, so okay, a length that is far greater than the dimension of a typical, that is 10 power 6. Nucleus dimension is 10 power 6, 10 power minus 6 meter. Okay, so in prokaryotes such E. coli, they do not have a defined nucleus, so the DNA, but still DNA is not scattered. So DNA being negatively charged, held with some proteins that have the positive charge in a region named as nucleate. Now the DNA in nucleate is organized in large loops held by proteins. Okay, so DNA in nucleotide is organized in large loops held by proteins. So in eukaryotes, this organization is much more complex. Now state of positively charged, that is basic protein, that is histones. As you can see, a positive charge acquires positive charge dependence of amino acids residue with the charge side chain. So histones are rich in basic amino acids, lysine, and arginine. Both the amino acid residue carry positive charge in their side chains. Now, histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Now, a typical nucleosome contains 200 base pair of DNA helix. Okay, nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. So nucleosome constitute the repeating unit of structure in nucleus called chromatin. Red like strain color bodies seen in the nucleus. The nucleosome in chromatin are seen as bits on string structure when viewed under electron microscope. The bits on string structure in chromatin is packaged to form chromatin fibers, okay, that are further coiled and condensed at metaphase stage of cell division to form chromosomes. So packaging of chromatin at higher level requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins. Okay, NSC proteins. Okay, so these are like the NSC proteins, NSC proteins, right? So NSC proteins. So in a typical nucleus, right? So in a typical nucleus, some region of chromatin are loosely packed, that stains light, are referred to as the euchromatin. The chromatin that is more densely packed and stains dark, these are heterochromatin. Now euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive.